Now that we've got xylem out of the way, the next step is going to be transport in phloem. So I'm using a pen now. Hope this is still something you can see easily. It's just that marker was getting really all scratchy and all dried out. And so if we talk about phloem, here's my tree again. One big difference with phloem versus xylem is that phloem has to go two directions. With xylem, we're concerned with only moving water up the tree. We want to go up towards the leaves. And since everything is evaporating out of the leaves, which are in the crown, that's the way it's going to work. We can really only go one way. But with phloem, we need to go both directions. You could have leaves maybe at this level, and the sugar that they're producing needs to go up, but it also needs to go down. Or maybe you have leaves up here at this level, and it only needs to go down. And so we need to be able to move the sugars that we produce in the phloem in both directions. And so we can't really rely on something like transpiration because that's only going to go from, well, up. And so phloem is going to rely more on just like diffusion. And so what's kind of neat to think about with phloem is that phloem is transporting sugar. And what's neat to think about is just that that sugar, it's made from carbon dioxide. Well, carbon dioxide is obviously a gas, and these sugars are a solid. And so the gas that is floating around these leaves is kind of being turned into a solid, which we then move around the plant. Well, the plant, once it gets the sugar, is going to respire it and turn it right back into CO2 again. So it's like we're making these solids within the cells out of thin air, but then as soon as they arrive to their target, they get turned right back into gas again. So it's like we're moving stuff around the plant, but when it goes from one location to another, it's like it's just disappearing to become a gas again. And so what that means is we're never really going to like fill it up. It's not like you're delivering a package to a house and then eventually the house is full of so many packages that no more will fit. Instead, it's like you're delivering food to a house and then they keep eating that food, and so it just goes away and they can always take more and more in. And so with phloem, we've got a few number of cells, and so we've got like a string of cells here. So we're going to have like a mesophyll cell and another mesophyll cell and another mesophyll cell. So let's say these are just all mesophyll cells. So mesophyll cells are just your regular old standard cell within a leaf. And so if they are inside of a leaf and they're just doing their job, that means that they are making sugar. Well, the mesophyll cells are attached to cells that are right around the vascular bundle. And so if they're sheathing the vascular bundle, these are bundle sheath cells. And you've heard that phrase before. We also talked about that with C4 photosynthesis. And so this may or may not be in a C4 plant, but these are just cells that are going right around that vascular bundle. And so we call them bundle sheath cells. Next, we've got a phloem parenchyma cell. So that means it's a regular old cell, just kind of a round, squishy guy, but it's a part of the phloem. Now we have a companion cell, and then finally we've got a sieve tube member. Okay, so the companion cell is right here, and then the end of the line is a sieve tube member. Okay, the sieve tube member is like the full-on main piece of the vascular bundle, and so the sieve tube member is going to move the sugar to what we call a sink. So a sink is going to be an area where there is a deficiency of glucose. The mesophyll cells are what we call a source. And if you can imagine what's being produced here, we've got glucose being produced at the source. The glucose could be used at a sink. So if we just kind of draw little dots to indicate the glucose molecules, we have a whole bunch of glucose in this area. We have very few down here at the sink. And so just think of this as like a concentration gradient. As we go from the source, from one cell to the next cell to the next cell, there are progressively less and less glucose molecules. And so kind of just using regular old chemical laws, the glucose wants to move over here to the sink. If the sink, which let's say it's the roots, or it's like a fruit that we're building, or a new leaf that's being produced. Let's imagine that new leaf, it's produced now, it's done. And so since it's done, it can start doing its own photosynthesis, and rather being a sink, it now becomes a source. It doesn't need this glucose anymore. And so it'll start making its own glucose, and so that diffusion gradient will go away, and the problem will just kind of fix itself. So we're going to talk about what each of these cells are doing, but really it's just we have a concentration gradient between a source where there's a lot of glucose, a sink where there is very little. The sink can change to become a source, or it can remain a sink, and that will either reinforce or take away that concentration gradient. And it'll kind of be a self-correcting process. So the sieve tube members are the ones that are actually going to transport it to a sink. And so the sieve tube member is, a cylinder, which is then going to be attached to another one, and it can go out through the rest of the plant. The companion cell is kind of a squishy living cell right next to the sieve tube member. And its job, if we kind of look over here, this is a good picture of what the two of them look like close up. This is our source cell, this is our sink cell. We've got our sieve tube member, the phloem, right here. And the companion cell is right next to the phloem cell, and it's just going to help transfer all of those sugars. Next to the companion cell is the Phloem Parenchyma cell, and again, it's just kind of a filler, helps transfer sugars. And then we have the bundle sheath cell, which is going to be the gateway. It's the barrier between the vascular bundle and the rest of the leaf. And so in this picture, this is showing a leaf, and it's showing the vascular bundle right here. The bundle sheath cell is around that vascular bundle, and the sieve tube members are in the middle. And so this is going to rely on this concentration gradient, and in between all of these cells we've got little channels where the glucose can travel from one cell to the next. So again, we got mesophyll cells, which are the source, sieve tube members, which are going to transport the sugars to the sink. In this picture they're going down, but they could just as easily go up if we had another sink that was above the level of this source. And again, it's going to be a self-correcting process because the areas that need sugar are going to change over time.